Okay, great. So today's think tank, rescheduled from earlier last month, is Matthew and Puchong. The title of the talk is Alpha CD226 suppresses diabetes development in the NOD mouse by augmenting Tregs and diminishing effector T cell function. Short bios on both of the presenters. Matthew is a biological scientist with the Todd Briscoe Lab at the University of Florida Diabetes Institute, or UFDI. Since 2019, Matthew's worked on improving Treg adaptive cellular therapy and evaluating the roles of T1D candidate genes in disease pathogenesis. And Puchong is an MD PhD trainee at the University of Florida. In 2023, he received his PhD to work on Treg conditional knockout of CD226 using the NOD mouse model for T1D, and he's currently finishing his final year of medical school. I'll drop the current paper into the chat, and we really look forward to this presentation. You guys can take it away.
All right, thanks for having us.
Sure.
So this presentation will be led by me. I'm just going to talk about a little of the background, and then I'm going to let Matt speak to the results of our recent paper. And so CD226 is a co-stimulatory molecule that is expressed on the surface of many immune cells, including T cells, where it provides a co-stimulatory signal that is associated with Th1 and Th17 responses. And the gene encoding CD226 was found to be associated with a gain-of-function risk variant associated with a number of autoimmune diseases such as type 1 diabetes, multiple sclerosis, and rheumatoid arthritis. And previous studies in our lab had shown that CD4 positive T cells that expressed CD226 on their surface also had high production of inflammatory cytokines like interferon gamma. And when looking at the regulatory T cell subset, or Tregs, that expressed CD226, they had an epigenetic signature that was associated with reduced suppression as seen in the demethylation epigenetic signature of FOXP3. And so what Matt did in the lab was he wanted to find out whether or not CD226 could be used as a good surface sorting molecule to select out the most suppressive population of Tregs. And typically, regulatory T cells are sorted based off of CD127 negativity. It's just another surface marker. But instead, he used CD226 to sort out this population. And what he found was that when you sort Tregs based off of CD226 negativity, these Tregs had a higher percentage of FOXP3 and Helios double positive Tregs that are associated with high suppressive function before and after expansion. And additionally, he took a look at, again, the epigenetic signature of the demethylated region around FOXP3 and found that before and after expansion in vitro, CD226 negative sorted cells had a preserved TSDR demethylation signal, showing that this could be a really important marker that you can sort suppressive Tregs from. He also did functionality studies where he co-cultured Tregs that were sorted based off of 226 negativity versus the conventionally sorted Tregs, and found that when you co-cultured Tregs with CD4 and CD8 responders, that 226 negative sorted Tregs were better suppressors of these responders as compared to Tregs sorted on the conventional strategy, showing that 226 could be an important surface marker when sorting Tregs for adoptive cell therapies for potential treatment of autoimmunity. We also had some animal studies working in parallel while Matt was doing his sorting studies, and some initial data that we found in the mouse model of type 1 diabetes, the non-obese diabetic mouse. What we found was that when you genetically remove CD226 expression from this mouse model, these mice had reduced incidence of type 1 diabetes compared to NOD mice that had intact expression of 226. And when you take a look at their pancreas, we saw that there was decreased inflammation of the islets of Langerhans that contain insulin-producing beta cells as compared to ones that had intact 226 expression. Interestingly enough, when we took a look at adoptive transfer studies into immunodeficient NOD mice, we saw that CD226 expression on CD4 cells was particularly important in their ability to confer type 1 diabetes into the recipients. And this kind of told us that maybe something within the CD4 compartment and CD226 expression was associated with disease pathogenesis. We took a closer look at a subset of Tregs, ex-Tregs, that at some point had expression of FOXP3 and suppressive capabilities, but for some reason or another, due to exposure to inflammation or some other stimulus, they lose their suppressive capabilities, and they maybe potentially contribute to the inflammatory process. And so we crossed our 226 global knockout strain with a strain that allowed us to permanently label Tregs along their lifespan. And what we saw was that when we reduced the influence or knocked out CD226 from the genome of these mice, we had a reduced population of destabilized ex-Tregs as compared to mice that had intact 226 expression. And this told us that 226 was somehow having some role in the destabilization of the cell population. We also generated a conditional knockout strain that only regulatory T cells in these mice lacked 226 expression in comparison to the rest of the mouse. And we still found that these mice that only lacked 226 on Tregs had reduced diabetes incidence compared to mice that had 226 expression throughout its whole system, telling us that 226 expression was having some inherent functional role in the disease pathogenesis in this mouse model. And not only that it is an additional, that overall that this could also be an important marker to look at for potential targeting with monoclonal antibody therapy.
So with these prior studies, we were interested in identifying a deliverable biologic therapy that could modulate CD226 activity in vivo. So to accomplish that, our study focused on anti-CD226 blocking antibodies. One of the first experiments we did to characterize the impact of anti-CD226 on T cells was evaluate its impact on T cell activation and subsequent proliferative capacity. So what we show here are dye dilution plots of mouse splenocytes treated in vitro with either an ice type control as a placebo or anti-CD226 blocking antibodies after 4 days of stimulation with strong TCR stimulation accomplished by using anti-CD3 and anti-CD28 stimulating antibodies or pathway-specific anti-CD3 and CD155 immunoglobulin, where CD155 is a binding partner for CD226. And what we note in both the CD4 compartments here on the top, as well as the CD8 subsets, is that anti-CD226 treatment of the splenocytes significantly reduces the proliferative capacity of those subsets. And to look at how anti-CD226 might impact cells in vivo, we administered either the ice type control or anti-CD226 to the non-obese diabetic mice. We administered 3 doses of the antibody intraperitoneally between 7 to 8 weeks of age into these pre-diabetic mice, and then evaluated their tissues ex vivo 5 weeks following that first injection. And what we observed when we looked at Ki67, which is a cell cycle marker and a marker of proliferation, was that limited to the effector memory subset of T cells in both the CD4 and CD8 compartments, there was a reduction in Ki67 expression, where CD226 is likely to be most highly expressed based on our previous data, suggesting that anti-CD226 may specifically limit the proliferation of some of those more effector-like cells. Beyond this, when we looked at the frequency of CD4 and CD8 T cells in the nod, we saw a slight decrease in CD8 frequencies in the spleen and pancreatic draining lymph nodes of mice treated with anti-CD226 relative to those treated with the ice type control. And based on all of our previous work, noting the impact of CD226 signaling on Tregs, specifically the destabilization of Tregs, we wanted to know how anti-CD226 might be impacting Treg function. So to accomplish that, ex vivo following treatment, we evaluated CD25 expression on Tregs in the thymus, pancreatic draining lymph nodes, pancreas, and thymus and spleen of these mice, where CD25 is the high affinity IL-2 receptor that's associated with Treg functionality. And what we note here is that mice treated with anti-CD226 had significantly higher levels of the IL-2 receptor, suggesting there may be differences in functionality resulting from treatment. So to take a closer look at how Treg functionality might be impacted by anti-CD226, we took splenocytes of mice that had been pretreated with the ice-type control or anti-CD226 and treated them ex vivo with recombinant interleukin-2 to evaluate phosphorylation of STAT5, which is associated with Treg fitness and functionality. And notably, what we saw in the mice treated with anti-CD226 is, to a greater extent, they were able to phosphorylate STAT5 in the Tregs, suggesting that anti-CD226 may be modulating Treg functionality. In vitro, we were able to evaluate the suppressive capacity of Tregs following a 30-minute pretreatment with isotype control antibodies or anti-CD226. So in the bottom, what you see are the results of a 4-day co-culture following pretreatment of just the Tregs with the antibodies. And what we note is that there's a reduction in dye dilution of the responder population that was cultured with Tregs pretreated with anti-CD226, suggesting that blocking 226 with an antibody may improve Treg suppressive capacity. Beyond this, we wanted to know how this might impact disease incidence in the nod. So first, we looked at insulitis, which is the immune infiltration of pancreatic islets, as Puchong previously explained. And what we saw is that mice treated with anti-CD226 5 weeks prior to when this histology was collected had a reduction in immune infiltration into these islets as compared to ice-type control-treated mice that had a greater extent of infiltration. As for disease incidence at 30 weeks of age, we noted that anti-CD226 treated mice between that 7 to 8 week window for treatment had greater protection from diabetes out to 30 weeks. And beyond this, we were interested in identifying the effect of anti-CD226 on some of these effector cells that might be driving disease pathogenesis in the nod. So we looked at CD8 T cells, specifically autoreactive CD8 T cells within the pancreas, specific for the beta cell autoantigen IGRP. And what we note is that after treatment with anti-CD226, these nods had a reduction in the frequency of these autoreactive CD8 T cells in the pancreas. In vitro, we evaluated the impact of anti-CD226 on autoreactive AI4 T cells using chromium release assays to evaluate the death of Th1 pancreatic beta cells plated in a dish. And we note here, across varying effector to target ratios, from 1 T cell to 1 beta cell up to 25 T cells to 1 beta cell. As that effector to target ratio increases, we see a slight reduction, but at these lower effector target ratios, we see that anti-CD226 offers some protection to those Th1 beta cells from cell-mediated lysis. To get a more granular understanding of what may be happening within these nods, we performed 10x genomics single-cell sequencing for both single-cell RNA-seq and TCR sequencing. And what we yielded in the pancreatic T cells of these mice were 15 unique clusters of T cells. And in our manuscript, we describe differences in gene expression associated with a reduction in Th1 and Th17 related vector genes. And what was really interesting when we looked at the TCR sequencing data and specifically annotated CD8 T cells reactive for that beta cell autoantigen IGRP was that mice treated with anti-CD226 had a reduction in the clonal frequency of those CD8 T cells reactive for IGRP, suggesting that in conjunction with our previous tetramer staining, that anti-CD226 is not only improving regulatory T cells, but maybe diminishing the effector capacity to kill pancreatic beta cells. So with this, our laboratory is continuing to investigate how anti-CD226 may impact human T cells, as well as evaluating other ways that we can modulate the CD226 axis and other co-stimulatory mechanisms as a deliverable biologic therapy for type 1 diabetes. And with that, Puchong and I would like to acknowledge many members of the Bresco Laboratory and PIs at the UF Diabetes Institute that have helped guide this research.
Great, thank you very much, Matt. Do you see any off-target effects in the NOD? I mean, I know that, CD226 plays a role in immune surveillance in AML or acute myeloid leukemia. Of course, since the NOD skid mice are really they already have this severely impaired immune system. You wonder if you might have to test for that in other model systems. But, for instance, like CD226 helps NK cells identify and destroy tumor cells. In other models, could you somehow devise some testing that would see how far out this effect with the CD226 might act without having any off-target effects?
Sure. So I think there's a couple follow-up studies that could be performed here, including seeing if repeated dosing impacts disease incidence, not to mention understanding the half-life of anti-CD226. But as you alluded to, CD226 is expressed on several other immune cell subsets, including monocytes. So our laboratory is looking into how CD226 signaling on those cells may be involved during disease pathogenesis, not to mention the impact anti-CD226 might have on that. But beyond that, I think before any sort of clinical trials performed, there would be to be an understanding of how anti-CD226 impacts other non-immune cell-related subsets, because we know CD226 is also an adhesion molecule found on epithelial cells and is involved in activation. So a more complex understanding will be required.
Yeah. No, I mean, it's expressed on a number of immune cells, right? It's like the NK cells, the B cells, dendritic cells, the hematopoietic precursors, platelets, T cells. So it's around. So yeah, I think that's smart to really dissect out all the different pieces. But this is really encouraging and interesting that you saw these direct effects with blocking it. I really liked your the visual you had in the paper. It was really very helpful to follow how it's actually blocking, how it's actually doing its blocking by enhancing Treg functionality and reducing the effector T cell proliferation at the same time. So kudos on that. Rami Crusoe, any ideas on mechanisms of the effect of anti-CD226 on IGRP-specific CD8 cells, like any evidence of deletion, greater susceptibility to AICD, or simply less expansion?
So specifically for the autoreactive compartment, there's some more work that needs to be done understanding the role of anti-CD226. We know from some of our initial data in the paper that anti-CD226 really suppresses the proliferation of those more effector subsets. So autoreactive T cells, specifically those reactive to IGRP, are more likely to be effector memory or central memory in nature. So CD226 is upregulated upon T cell activation. So the most activated T cells would be those expressing the most CD226 and most likely to be impacted by anti-CD226. So whether there's an impact specifically on TCR specificity is unclear, but we would speculate that based on the nature of autoreactive T cells and their more effector nature, that anti-CD226 would impact those cells at a higher bias than naive T cells.
Good answer. Anybody else? Hands, you can just unmute yourself here. Eddie James, do you have any concern that anti-CD226 might impair protective immunity? That's sort of what I was getting at a little earlier too, right? Because its
Right.
role is immune surveillance, especially in like AML.
So I think for any immunomodulatory therapy, there needs to be a careful titration, right? That we don't have concerns with opportunistic infection or downstream the emergence of cancers. So as compared to completely immunoablating therapies, anti-CD226 is a bit more targeted and sparing some of those naive cells while impacting the proliferation or cytotoxicity of effectors. So, but more work has to be done to develop better therapies for type 1.
Yeah, it's great. I just wanted to go back to the SNP that you referenced in CD226, I think it was the RS763361 cytosine, results in sort of this GLY307 serum missense mutation, and that's linked, right, to genetic susceptibility to type 1 diabetes. How prevalent is that in the whole type 1 diabetes, sort of patient, those who go to stage 3 to diagnosis of type 1 diabetes? What % of people have that SNP? Is there any kind of data out there?
Yeah, so the specifics on the odds ratio, I don't have at the top of my head, but our laboratory did publish a paper discussing multiple genetic risk variants in type 1 diabetes, CD226. So it's not a rare variant patient population.
Okay, that's great. It's good news if this really, if this therapy progresses and it works, then it would serve many people.
One thing I'll note about that particular variant is that we hypothesize that it contributes to increased CD226 activity. So we'd hypothesize antibody might have an even better effect in yielding a response in those patients.
Nice. Looks like someone dropped in the chat. 0.47 is the number of patients that have that susceptible SNP. Okay, 47%, I guess. So that's great. It's a good target.
Yeah, so I'll say that what that implies is a very common variant, but it also doesn't necessarily mean that it wouldn't impact people with the wild type allele, right? It just means that for that singular locus, it has some small role in and of itself as a risk locus, but certainly a variant in that is somehow leading to more activity and probably more effector function of some of these cytotoxic cells or dysregulation within the Treg compartment. But it is a very common allele in the population.
That's good in some ways, right? It's for this type of therapy. We have some exciting talks set up for next week with the University of Florida Diabetes Institute. We're going to showcase several of their research groups and, kind of shine a light on what is going on at UFDI, which is a lot. As well, I understand that there's going to be some really exciting presentations coming up from UFDI at NPOD. So great, really great news for UFDI, and it's exciting times for that whole institution. We'll be really excited to keep on watching and showcasing your efforts. Thanks again, everyone, for joining, and have a great rest of your day.